Good afternoon, everyone. We will talk about leukemia. And 1 second, please. Sorry. Yes. So we are going to talk. Actual topic that we are going to discuss as a part of your syllabus is the therapeutic strategies for cancer stem cells. And in the previous version of this syllabus, we had leukemia as well to read as a part of cancer stem cell biology, the third unit. So I will just glance upon these so that we will have some knowledge, and then we will move on to these topics of therapeutic strategies. First thing before going, I would want to also discuss is some of the authors, or in the older version of the books, many of the books, pluripotent would mean, in that cases, they meant that it means it is anything that is multipotent was or used to be. Some people use it still in the older generation, probably they use it wrongly. For us, we are very clear that totipotent means it can give rise to a complete organism. Pluripotent is all types of cells but not complete organism. Then we have multipotent, which can give rise to multiple cell types. And then we have unipotent stem cells, right? They can give rise to only one type. So we need to keep this terminology, we need to use this terminology very accurately. And if we look at any of the, we also discussed about this, adult stem cells. Adult stem cells will give rise to, occasionally will undergo asymmetric cell division. They will give rise to transit amplifying cells, which will then ultimately, give rise to progenitors and ultimately the differentiated cells. So if we think about this diagram, which is one of the prescribed syllabus, one chapter. So the authors of that particular chapter may have meant it differently, or it could be a typo. So anywhere, normally this is the where adult stem cell will give rise to the tissue cells. But anywhere this kind of blockage can occur, and then they can probably become cancerous, right? And that is what we have been also discussing. We discussed those things when we were talking about cancer stem cells in that particular unit. So what is leukemia? Leukemia is a malignant cancer, we say. Normally benign is somewhere located within one particular area. It is slow growing and not so aggressive. But once it moves to different locations, then we have tumors in other places, then we will say it is malignant and metastatic and so on. But because leukemia is the cancer of the liquid tissue, that is where we also discussed about liquid tumors, right? This is a liquid tumor. That is the cancer of the liquid tissue. So this is one of the problems, is because there is a block of maturation. So the hematopoietic stem cell should give rise to ultimately blood cells and lymph cells. Somewhere during that process, the differentiation is not incomplete, that maturation is not happening. And then we were ending up with so many immature white blood cells, which again is a sense, as we discussed about tissue number homeostasis, there will always be dying of cells, and new cells are produced from the resident stem cells, adult stem cells. But here, when these cells die, and the adult stem cell here, hematopoietic stem cell, would produce cells, but because there is a maturation block, these undifferentiated cells are produced, or immature cells. The need is still there because these are actually not functional. So then the stem cell try to compensate and keep on producing more and more. And these are white cells, and that's where the term leukemia, leuke, is about white. The color is between somewhere red to creamy white, so white blood. It's something like that. Leukemia means white blood. So a normal blood is filled with so many red blood cells and these white blood cells, whereas in leukemia you have too many of these immature white blood cells, and that's why the coloration of the blood also changes. So this is the normal, whatever I discussed. Hematopoietic stem cell would give rise to the progenitor cells and then the mature cells. But somewhere a transformation event has occurred. Transformation is a normal cell transforming into a cancerous cell, where they keep on producing leukemic blast cells, which are non-functional, and that is a burden to the body and the tissue itself. So there are multiple pathways that could lead to leukemia. Impaired differentiation is one part. Increased cell survival. The cells should die after a certain time period. The cells have cell turnover, which means they are alive for, say, for red blood cells, it's about 3 months, I think, and then they die. Right? So when they are living for too long, then that is one of the increased survival is one of the problems. Increased self-renewal. These are some of the signaling pathways that we discussed. Increased proliferation and genomic instability. All these could be involved or could cause or be involved in the formation of leukemia. One of the things that we will discuss is a little bit about this. There are multiple types of leukemia. We don't have to mug up those things, but if a blockage happens somewhere here, they say pluripotent, again, that is wrong, right? We will just say it as multipotent bone marrow stem cell. If the maturation is blocked here, it is AML, acute myeloid leukemia. If it is after promyelocyte, there is a blockage here, then we have as PML, promyeloid leukemia. And then there are other, after myelocyte, if there is a blockage, then we can say CML, chronic myeloid leukemia. One of the reasons is reciprocal translocation. Reciprocal translocation. That means it is a chromosomal aberration, meaning that there are chromosome number 9 and chromosome number 22. There are two genes, one is ABL and the other one is BCR. Sometimes they call it as a fragile chromosome or fragile spot, where the chromosome could be broken. These two chromosomes could be broken like this, here and here, and these parts are exchanged. That's why we call it as reciprocal translocation. And now we have a new fused gene formed. We had ABL and BCR, and now on chromosome number 22, which is referred to the modified one after reciprocal translocation, we call it, there is a name given to it, it is called as Philadelphia chromosome, and it has a fusion protein like BCR ABL. So this was producing one protein, this was producing another protein, and now after the formation of Philadelphia chromosome, it forms something like this. Okay, that's a fused protein product. It means it is compromising 2 points. One is we don't have a ABL now, and we don't have a BCR, and we have an unnecessary product here, BCR ABL, which is twice or thrice the complication. So what BCR ABL actually has is it has an ATPase binding domain, and it has some other substrates which will phosphorylate the substrate. So BCR ABL, the fused protein or the hybrid protein, is this one. It has a pocket for ATP, and if we want to we should be able to inhibit the activity of this particular BCR-ABL. That is one of the reasons for causing the CML, chronic myelogenous leukemia. So what it does is it binds to ATP and phosphorylates. It's a kinase. It phosphorylates the substrate. And now we have a substrate because of which will bind to its effector. This is activated because of phosphorylation on the tyrosine residue. Tyrosine has an hydroxyl group. On that, phosphorylation would have occurred. And now the substrate is active, and therefore the effector gets active, and then that results in chronic myelogenous leukemia. So unless we block the activity of BCR-ABL, we will not be able to control or regulate this CML. So there is a drug called as Imatinib. Imatinib is a drug that binds specifically to the ATPase binding site here. And if that site is occupied by Imatinib, there will not be any more ATP binding here. Therefore, the substrate will not be phosphorylated. And then the effector, we are blocking the formation of CML, chronic myelogenous leukemia. Okay. So, but the problem is in treating leukemia, for example, is there are always mutations that are possible. Every cancer is like a hot pot for mutations because they're growing rapidly. There are a number of cell divisions, and there is inherently genomic instability, because of which there are more and more mutations that are happening. And if the rate of mutation is high, the probability of increasing beneficial mutations to the cancer, mutations beneficial to the growth of the cancer, will also be highly probable. And so, and that is one of the reasons where they may develop some mutations within the kinase domain, because of which Imatinib is no longer useful to the, or cannot inhibit this fusion protein. And also, using Imatinib is per se not a cure. It is kind of maintenance. As long as one is using Imatinib, it will, the BCR-ABL fusion protein is inhibited, and when you stop doing it, it will recur. So, and the second problem is that when we are constantly using, then we will be selecting for BCR-ABL genes that are no longer inhibited by Imatinib. So those are the problems associated with leukemia and treatment of leukemia. So I will skip this. It is all just discussing the other one where another fusion protein is formed that is called as PML, promyeloid leukemia, RAR alpha. This is another one that is involved with another type of leukemia. We will discuss about RAR in a while. Okay. First thing is we want to talk about what are the strategies that we have to get rid of tumors. So if we say that this is a tissue and there is a tumor, how do we kill it? How do we get rid of it? The body has a problem with associating with it. First thing is surgically we remove it, surgery. If we are doing it at very early stage, the probability of success rate is very high. But in an advanced stage, when the tumors are forming in different parts of the body, how many tumors can we remove? And we cannot be sure that there will not be any more tumors that will come by. So at least in an advanced stage, surgical removal is not so useful. Probably we can do it so that it doesn't influence immediately. We can prolong the life of the person. The another strategy is to inhibit the proliferation. We have a tumor, there is a cell in it. If we can control its proliferation, reduce its proliferation, then the growth of the tumor will also reduce. That is another strategy. And the third is induce apoptosis. Somehow use some drugs or some chemical components or biological ones, so that this cell undergoes cell death. And that's why we can probably, anywhere these cells are, they might probably undergo apoptosis and die. The fourth one is to differentiate. So from adult stem cells, we have TACs, and then we have the terminally differentiated cells. So somewhere, because of these blocks, these cells are not differentiating. If they differentiate, they would have a certain rate of certain duration lifespan, and then they would have died. Because of these blocks into maturation, they remain undifferentiated, immature cells. And if we can somehow prevent these blocks, then they would undergo differentiation, and they will undergo the normal process of lifespan and then death. That is the other strategy. Trying to remove these blocks by some mechanism, so that the cell can differentiate or be forced to differentiate and then die out. In the olden days, they used to have something called as cauterization, where the tumors, wherever they're present, they would have burnt a iron rod like this, and then they would have put it at the site of on the tumor. You can see, I think in Egypt and many places, they did practice it until some 500-600 or 300 years back as well. It is even now useful in modern one, but they do it differently. Cauterization is using this red hot iron, or we'll just say it as heat, to kill the cells. And there are two advantages. Say, for example, if somebody was using surgical removal, and in those times where we didn't know how to control the blood flow, I mean, when we cut something out, the blood will drain out and the person would die, right? But nowadays we have technology, we have blood transfusions to control those things, and many things to do. So surgical removal was not as easy in those times. Cauterization does one thing: it will kill the cell, and it will seal that gap also. It's not slicing the blood vessels; it's rather sealing it, so there will not be as much blood loss. If you look at laparoscopy videos that are present on YouTube, you might want to check it out actually. So they put the small rod-like things into our abdomen through some sutures, through some holes, punctures they would make, and in using a camera, with the camera and these 3 tubes would have been inside the abdomen. That means you are not cutting so much of the abdomen, only 3 holes. And then these would be having some pinchers like this, and they would get hot. They would hold a blood vessel, for example, and then they will put in charge, because of which these will become very hot, and they would have sealed the blood vessel. So there would not be any more blood loss from those places. It's interesting to know about the history of surgery as well. We had our own ancient father of surgery as well, right? Okay, so for cancer therapy, what we choose is, as we said, elimination of cells. And when we talk about the cancer stem cells, how we can approach it is the traditional therapies that we know of: radiation therapy, chemotherapy, anti-angiogenesis, that is trying to prevent the formation of blood cells so that the tumor does not receive enough oxygen and nutrition, because of which they would die. So radiation therapy most of the times, or chemotherapy, is to block or break the DNA, because of which, if the cell undergoes cell division, it will form non-productive cells, and they would die immediately. We are increasing the rate of death of these cells. And chemotherapy is also similar, almost similar ones. There are different chemotherapeutic approaches. There are multiple of them. And then differentiation therapy, what I explained to you is that we somehow try to remove the block from maturation of the transit amplifying cells into differentiated cells. If we do that, then the cell should go this way. And for the cancer stem cells, if we want to do it, we should try to use inhibition. We will discuss in the following slides. So this is the general thing, is we use radiation so that the DNA is broken and the tumor cells will break. There are normal cells and there are cancer cells. Normal cells, if they get DNA damage, they would stop growing. They would stop growing. They will perform all the repairs that should be done, and only after the repair is done, they have tightly regulated cell cycle. After the repair is done, they would undergo mitosis. Whereas a cancer cell does not have this strict regulation. It will just keep on dividing, because of which there are severe damages, and that's why these cells would die. Anti-angiogenesis, we know VEGF, which is important for the formation of new, for angiogenesis, formation of new blood vessels. The cancer cells usually do produce this VEGF, which will activate and the endothelial cells to form more blood vessels. If we can somehow block those, so VEGF is the one that is going to bind to the VEGF receptors and then stimulate the endothelial cells to form blood vessels. So we can do multiple ways of how to use anti-angiogenic ways. One of it is to remove the VEGF by adding antibodies, monoclonal antibodies against VEGF. So when the antibody is bound to the VEGF, VEGF cannot interact with the receptors. So we can probably do it that way. There are also other strategies where we can add antibodies against the VEGF receptors, so that when the antibody is bound here, it is no longer available for VEGF to bind to the VEGF receptor. There are also other small molecules. Some of these, the yellow ones, indicate the names or maybe the commercial names of what they do. Small molecules can act on the inside of the VEGF receptors. Even though VEGF is bound here, it will prevent from this phosphorylation or the post-translational modification, or block the binding of the substrates to it, thereby preventing the angiogenesis. So the problem with cancer stem cells is that the cancer stem cells are different from the rest of the cancer cells. They are also different from the normal cells, right? Cancer stem cells and these two are part of the tumor, cancer stem cells and the rest of the cancer cells. These cancer cells are the aggressively growing ones. So when we induce the radiation or so, majority of these cancer cells will die. But the cancer stem cells, they do not die. Mostly because majority of them are quiescent, meaning that they are kind of dormant. And if they are dormant, they have sufficient time to repair the broken DNA, even if they are exposed to the the radiation, say. They have time enough to repair the DNA. And while the rest of the cancer cells would die. And when the radiation is over and after some time, these cancer stem cells will regenerate a new tumor. Similarly, if you are using some chemotherapy, which we are using small molecules, they may enter in. But as we discussed, many of the cancer stem cells also have high expressions of APCG2, ATP binding cassette efflux pump, right? I hope you remember when we are characterizing how to characterize site populations, maybe. So they do have high amount of efflux pumps, because of which Even if a drug entered into the cancer stem cell, because it has high ABCG, it will be pumped out, thereby protecting the cancer stem cell. Okay, so these are at least two of the mechanisms that are resulting in the resistance of cancer stem cells to drugs. Then comes the immunotherapy. Immunotherapy is quite popular because they can bind to different types of molecules, and if mostly they are about the receptors. So if we have known, as I was discussing about cancer stem cell, we discussed that CD133, CD44, they are the popular cell surface markers that are present on cancer stem cells, CD24 also in some cases. So if we can identify these cell surface markers that should be highly specific and should be in higher concentration, and if we can do that, and then if we can generate antibodies against these cell surface markers, then we can target the cancer stem cells specifically. Targeting cancer stem cells specifically is the goal of cancer therapy, isn't it? Because every time they undergo chemotherapy or radiotherapy, the patient actually suffers a lot. It takes a big toll on them. They have to undergo a lot of pain for a prolonged period of time in multiple rounds, second round, third round, or last round. They keep saying those things. They lose all their hair and they become very weak with the suppressed immune system, and many things are there. Okay, this is how the immunotherapy can be used if you want to target. But the limitation is that we have at this stage is still our inability to or be specifically able to kill these cancer stem cells. If you remember, we discussed about carbonic anhydrase when they grow on the hypoxic conditions. Carbonic anhydrase will allow the cells to grow even in reduced or even in acidic pH. Warburg effect, we discussed those things. So if we want to use immunotherapy, or we discussed carbonic anhydrase 9 is prevalent or highly expressed in these cells. So if we want to do those, we can use multiple ways. This has to do with antibody engineering. That's a good topic as well. Antibody engineering. An antibody is something like this. It has disulfide bonds attached like this. We have 2 light chains and 2 heavy chains. The big ones here are the heavy chains. You might be studying it right now. And these are the light chains that are present. And the region here, that is where using which they would bind, that is important for specificity. They would bind to their target epitopes. Epitope is the region on the antigen which is bound by the antibody. So each epitope will have one type of antibody. And we also have these big, large constant regions which are important to trigger the immune cells to do whatever it is doing, like killing the cell, for example. So there are 2 components to it, to an antibody, if you see it in terms of function. One is to have specific interactions, identify the epitope. The second thing is to activate the immune system and then either kill it, degrade it, or take it up and do some form of elimination from the body. So we have an antibody typically is like this, and that means it has 2 sites for binding specificity. It is bivalent, we call it, because it has 2 regions to bind to the antigen or epitope. So antibody engineering is about how you are modifying it. What if I attach a toxin molecule here? So, and assume this is anti-CD20 CD133, for example. If this antibody is against CD133, it would bind. CD133 is here, say, and the antibody would have been bound here along with the toxin protein, and then it could be taken up, internalized like an endosome, and the toxin protein would be released, killing the cell, right? That is antibody engineering. We have attached this toxic protein to the constant regions of the antibody, right? So that is one strategy. There are diphtheria toxin. I think a beta fragment of diphtheria toxin is useful for this. In this case, some people have already tried at least doing those kinds of antibody engineering. We can also give radionuclide attached. The radionuclide is attached to the antibody, so it would be taken up by the cell, and the radionucleotide is delivered into the cell that is specific. The specificity is determined by the antibody. Here it is against carbonic anhydrase. We can also use cytokines. We can also do something else, that is bispecific antibody. This is truly taking it a little further. As I said, one antibody will have specificity for one epitope. What if we constructed an antibody we engineered an antibody in such a way that it becomes bispecific? That means we have one antibody made from two different parts, two different antibodies. We have fused them like this. So one region is specific for one type of epitope, and this one is specific for another epitope, right? So that is bispecific antibody. If we could make that way, one part specific for carbonic anhydrase and the other part is specific to a cytotoxic killer cell, then you are bringing the killer cell near to the toxic cell, to the cancer stem cell, and that could be killing it. Okay, so there are multiple strategies how you can use antibodies. This is antibody engineering. So one should be strong in immunology, especially if you are venturing into any of the animal systems, or mostly about the clinical side. Every clinical side is it is about immunology, the body's ability to heal itself, to clear off the disease itself, and how we can use it, or how we should be considerate of the effects of immune system, right? So be strong in immunology as well if you are going into the clinical or medical side of the biology part. And then we have some cancer stem cell specific therapeutic approaches, which we have already discussed. And just to summarize, we discussed about 3 signaling pathways: Notch signaling pathway, Wnt signaling pathway, and hedgehog. And we know the different the doers here: NICD, beta-catenin, GLI213, the activator. So we could have some kind of drugs made against the proteins that are involved in these signaling pathways, where we can say if we can have a gamma secretase inhibitor, so that the gamma secretase, if it is inhibitor, there will not be any production of NICD, right? And then we could probably these genes will not be turned on. Similarly, we can also have the antibodies against Wnt, antibodies against Frizzled, or inhibitors against several of these proteins, including beta-catenin, so that these genes will not be overexpressed at least. And the same way we can have antibodies. If they're on the cell surface, it is easy to develop antibodies against them. If they are inside the cell, then if it is inside the cytoplasm, antibodies cannot reach them directly. We can also use we can also use BMP and others that can increase the differentiation pathway so that the cell that is blocked from maturation could get differentiated and then it would die out. If we ever can become that there is a specific cancer stem cell antigen which is not present in any other body, say for example something like a fusion protein that has been formed, okay, say a fusion protein is there, because when you talk about Wnt, gamma secretase, Wnt, and all these proteins, they are present in normal cells. They have a normal function to do as well. So we cannot make vaccines against those proteins because then the immune system will go havoc and kill many of the cells and the individual would die. But if we can ever in the future know if there is a specific cancer cell, a stem cell antigen, it should not have been found in anywhere as cells. If we can ever do that, then probably we can generate vaccines against it, so that using that peptide we can generate vaccines which when we give, the individual will raise antibodies against it, and anytime such a cancer stem cell arises, that cancer stem cell will be targeted by the immune system and eliminated. But we are very far from it, and in my personal opinion is that I would doubt if we would be able to find anything cancer stem cell antigen. Maybe somewhere out of the many hundreds of cancers, there could be one type of cancer in which something like, say for example, we spoke about leukemia, BCR-ABL. It is an intracellular protein, so it cannot be used as an antigen directly. But if there is any cancer in which a fusion protein like that is formed, the fusion protein is only found in those cancer cells and nowhere else. So there, at that time, in such cases, we can develop cancer stem cell vaccines. Okay. And there are many stem cell inhibitor proteins that people have been using these names. We don't have to mug up, but try to know that this is how we can attack. Anytime when somebody gives you a situation, you should be able to think of strategies about how best we can an approach. We should be able to see the approaches of how we can probably treat or solve that problem. So you can look at this table. I'm not going to discuss much about it. There are also other therapeutics using the molecular techniques like the RNA interference. We discussed a little bit about it in our previous course, molecular genetics, where there is a formation of RISC complex. The dicer will then cut the DNA, the mRNA, and then we'll have cleaved mRNA, because of which the protein is not produced. Say if we know that a particular mRNA is produced in excess, whose product is the one that is causing the cancer? And then we can design these short interfering RNAs and then target them to the cancer stem cells, and they would then eliminate the mRNA, because of which the protein is not produced. Therefore, we might be treating the cell or killing the cell or reducing its growth. How do we give it specifically to the stem cells? We have normal cells, we have cancer cells, and we have cancer stem cells. What our target is cancer stem cell. How can we specifically deliver to it? Okay, we can design a fantastic chemotherapeutic agent or a DNA or RNA, and we want that cargo to be delivered to the cancer stem. In that case, based on the chemical properties of the compound, we can make either liposomes or micelles and deliver them. If it is a hydrophobic agent, we would put it in so that it will be within the hydrophobic region. This is a bilayer. Liposome has a bilayer. Micelle is a single layer. Its micelle would be the hydrophilic outside and the hydrophobic interior. Liposome, on the other hand, will have two layers, the lipid bilayer like the cell membrane. This is one layer, and this is the other layer. So we can have, if it is lipid soluble drug, we could use micelles, or we could probably put it in the hydrophobic region of a lipid bilayer like that. If it is crystals, you can have them inside, and DNA is also polar, which can be put inside. The point is, how can we make it go to the target stem cell? For that, we could use, say, if there is a receptor for this particular, a peculiar receptor is present on the cancer stem cell, then we could use a ligand which will go and bind there. And then there is a probability that the liposome would fuse with the cell membrane and deliver the cargo into the cancer stem cell. We can also use antibodies so that that will bind to the specific targets on the cancer stem cells, so that it will increase the chances of the liposome binding to the cancer stem cell. So we may also need to have some protective layer against immune system. Immune cells will degrade anything that they find is not useful or foreign. And in that case, we may need to have certain components that may give protection from immune system. That would be the ideal liposome drug delivery. You have a course on drug delivery, I think, in the future semesters. There you probably would learn it much more in detail. Okay, I want to have a brief discussion on the differentiation therapy. In one of the experiments, Pierce and Wallace, they showed that if there is a squamous cell carcinoma, this is, say, tissues, this is the tumor, and they can become, this is the, on the skin, this is the keratin layer that is present on the utmost surface of our skin. They have taken a tumor and they have labeled it with radio-labeled with DNA, radio-labeled the DNA, and they put it under the skin of mice or so. And what they have noticed is, after some days, in this keratin layer, they found radio-labeled radioactivity, which means that these cells, cancer cells, have become part of the keratin layer. Sorry. That means the cancer cells are dying and becoming a part of the keratin layer. So what this proves is that cancer stem cells can can differentiate and form something else, which are relatively not so dangerous to the host organism. You know about Lance Armstrong, I guess. Sorry. There is a tournament called Tour de France. I think they go around some 256 or so kilometers through the Andes mountain region. Excuse me. So they travel through these mountain ranges. Some are very steep like this, some are plains, and some you have to climb uphill. And Lance Armstrong is an American, and their team won 7 times, I think. But later, he was accused of drugs, doping, and then he eventually admitted as well. Anyway, this is a very famous tour, the event, and then they struggle. That is one of the most competitive ones, in the sense demanding high level of fitness. Lance Armstrong is also known for something else, that he had teratocarcinoma. It's a teratocarcinoma, a tumor in the testicles, and then he defeated it in a way using differentiation therapy. Okay, I think so. So what is differentiation therapy? We'll have a brief concept about it, and then we'll stop. This is about somehow identifying or somehow blocking the signaling pathways that maintain the stemness. Okay, if there is a cell, we have stemness of self-renewal, right? I think the next slide will show it better. Oh, this is the slide. 1 second. There is a stem cell, and the stem cell gives rise to transit amplifying cells, and they will give rise to differentiated cells. What happens? There is also certain degree of stemness. You can think of this one has, whereas stem cell has high rate of self-renewal capacity, transit amplifying very less, but there is little bit. That's why they amplify in region numbers and then divide into differentiate into terminally differentiated cells. If the block is happening, if the maturation is blocked, then the transit amplifying cells will increase in number, and there are no differentiated cells or functional cells, because of which the differentiation or asymmetric cell division is more. Thereby, we are increasing the number of these non-productive cells, and that is tumor. So the idea is, what if we can block these stemness? If we have blocked the stemness or the self-renewal properties, and that means if you have a stem cell and it divides, it undergoes asymmetric division, one of the same cell type and one differentiated cell. If we somehow, by blocking signaling pathways, if we force it to divide, or we just block the signaling pathway, then what would happen? Both the cells would become terminally differentiated cells or non-stem cells. Then in that case, in this case, how many stem cells are left? 0. Whereas if you consider this, how many stem cells are there? There is 1. So the goal or basic concept of differentiation therapy is somehow, if we can target the stemness itself, then we can probably allow the differentiation to happen and eliminate the cancer stem cells, right? So teratocarcinoma and leukemia, people have tried this differentiation therapy, and there has been considerable success. For leukemia, they used retinoic acid, and it binds to PRL or PML and RAR, leading to that has resulted in differentiation of these cells into terminally differentiated and eliminating, to a certain degree, differentiating the leukemic stem cells. What retinoic acid does is, okay, retinol, that is vitamin A, which is reduced and it forms retinol, and then retinol dehydrogenase, I think it is oxidized, I guess. Then it forms retinoic acid, and then eventually it forms the oxidized product. The retinoic acid binds to the complex here, retinoic acid receptor. This should be the receptor, yes. When there is no retinoic acid, the RAR and RXR, they form a complex, and then they block the, they are not, histone deacetylation happens, because of which there is a repression of transcription on certain genes that are involved in differentiation. When we add retinoic acid binds to retinoic acid receptor, and because of which the coactivator binds here, right? Coactivator is recruited, and that will allow histone acetylation and thereby activation of transcription. And then the cell will undergo differentiation. So they hopefully we will not have any more cancer stem cell left. We don't have to go through all this pathway, but the classical and the non-classical pathway, I think I have one more slide. So retinoic acid, that is retinol, where is it? Yeah, retinol and retinal, and then retinoic acid, which will be taken up inside, and it can bind to this RXR RAR thing, and it will recruit several deactivators and histone acylases, thereby allowing the transcription of several genes, and it promotes cell growth. In the classical pathway that we know, the retinoic acid binds to these complex, and anytime you find this RE stands for response element. Okay, RAR here, at least RE is for retinoic acid, and RE is for response element. So in this case, when RAR is produced, it binds to the retinoic acid, RAR RXR complex, and will allow it induces the transcription of genes that are involved in differentiation and inhibits growth. Okay, well, this is the pathway, the classical pathway, at least in one case. There is some success with retinoic acid, but it is still not so popular. Because when we see in humans, the retinoic acid treatment is not as consistent in case of treating teratocarcinoma. With test organisms like the mice and rats, I think they have seen some success, but it's not as much in humans. Retinoic acid treatment is quite successful if there is any. It is better in leukemic conditions. Yeah, we'll stop here. This is a teratocarcinoma, which can be treated because it forms, say if it formed near the testis or other things that we discussed, testicular cancer, carcinoma, then you can just remove those and without affecting other tissues as much. Okay, I hope you understood the essence of the differentiation therapy and the different pathways and the ways of how to treat the cancer stem cell based or models, you can say. There is one thing that you have to remember is we need more research before anything of this can actually happen. Majority of the pathways are shared between normal cells and stem cells. We have normal cells, we have adult stem cells, and we have cancer stem cells, right? And if you see any of the molecular say signaling pathways involved or cell surface markers, they are shared at least among two of them. So anything that we found in cancer stem cells could be in adult stem cells, could also be in normal cells. Then finding a protein that is highly specific to cancer stem cell is the huge limitation that we have at this stage. And that is why people study more about it, and then we'll try to do better things. And hopefully we'll have better ways of regulating or treating the cancers in the future. Okay, I'll stop here. Any questions?